Assalamu alaikum wa rahmatullahi wa barakatuh. I'd like to thank the organizing committee for inviting me today to present. And actually my lecture it will be like a starter. مقبلات حتكون for the guys presenting after me. So, I don't know, I like to move around, but today they are fixing the phone here. Anyway, so today I'm gonna take a history about the diagnosis of viruses starting like we'll take a history starting from right the beginning when the viruses are, arises, and then we'll go through all different types or techniques that involve in diagnosing viruses up to PCR. So the history started when Pasteur tried to find the causative agent of rabies. And he wanted to see that agent under microscope, but unfortunately he couldn't see this agent under microscope, which is, rabies virus. So after that, Charles Chamberlain invented a filter which can pass the water and prevent the bacteria from passing with these waters. After that, Dmitry Ivanovitsky, he used this filter to study the tobacco mosaic virus. He crushed this virus with the water and hoping that he would prevent this virus from hoping to prevent this virus from passing through this filter, and then he can study more on this virus. But unfortunately, this virus could pass with the solutions because it's tiny, very tiny and smaller than the pores for the bacteria. Then they realized, Ernest Williams, he realized that this agent only survive or multiply only in living cells. For that reason, he grew an influenza on fertilized chicken eggs. So at that time, they realized that this new infecting agent only grow in living cells. Then Ernest Ruska and Max Knoll, they invented electron microscopy, which this is the filter which was used by Chamberlain at that time to prevent bacteria and hoping to prevent this small new agent, which is viruses, from passing through the filter. So why we should diagnose viruses? So simply because some of them are life-threatening infections like HIV, and to monitor and enforce vaccination strategies, to identify outbreaks. Also, some viruses are not that important, but still for epidemiology, they are important for us to know how many people are infected with these viruses, and to identify the new agents. So, diagnosing the viruses depends on two categories. Although in some books or some websites they make it 3 groups, but I followed the 2 groups. Oh, shukran. Thank you. Now I can move. So, either isolation of the virus from the samples, which will be in 3 ways, either from embryonated eggs and different sites in the egg, so they are like allantoic or olear sac or the chorioallantoic membrane. So every virus or each group of viruses, they prefer to grow in certain sites in the embryonated egg. Sometimes they use animals. Animals can be used like dogs, mice, rabbits. But sometimes, it will be problematic, especially where to keep these animals. They need special care, So they jump to the tissue culture, which is more easier. For me, I work in tissue culture. It was very easy for me, especially in hepatoma cell lines, and you can work. The only thing that you are worried about is contamination. Sometimes when you get a contamination in these kinds of cells, then you have to get rid of all these cells, clean incubator, but otherwise they are very straightforward. just what you need, just flask, trypsin editor, and just keep them in the incubator at 37 with CO2. And then once they grow, just hatch them and spread them in new flasks. The other direct examination of viruses can be direct examination. One of them is the electron microscopy. Electron microscopy, it was very good technique at that time, but the problem with electron microscopy that you need experience. I worked on it for a couple of months, but I found that it's very difficult because you are dealing with very tiny, square and about more than 100 smaller squares, and you have to examine in 15 minutes. If you get nothing, then sample will be negative. Okay, imagine this sample contains only small viral load, so we'll get false negative. So if you are lucky, then if you have more viral load in this sample, then can you diagnose it as a positive? And according to the British protocol, you examine only for 15 minutes. If you get things, positive. If nothing, put it negative. And the only thing that we found it from the electron microscopy that we can get the shape of the virus. So some viruses have very characteristic shape, like rabies. We call it bullet-shaped virus, which is like the bullet. And then detection of the viral antigen, immunofluorescence. This is my own work. I use hepatoma cell lines and transfected with hepatitis C, with the Replicon at that time, using the Replicons for hepatitis C, and injected, stain it with the monoclonal antibody against NSC3, which is one of the proteins of hepatitis C virus. And as you see, the stain here in the cytoplasm in the cells, because the virus usually multiply and the cytoplasm and doesn't move to the nucleus because it's RNA virus. So most of the replication, it will be in the cytoplasm of the cells. And then serology, which is ELISA. Everybody knows about it. It's an antigen and antibody reaction. It's a good technique used widely everywhere, and we are happy so far, but we have a problem, early infections. So this or when the period, when you get someone has a new infection, then you may diagnose it as a false negative, although he's infected, but still antibody is not there, so you will diagnose him as negative. Cross-reactivity, this is another problem with ELISA, especially in GISA, because we have dengue virus, and if the patient is infected with dengue virus and you diagnose him, investigate him for hepatitis C, you may get false positive, because dengue and hepatitis C, both of them from the same family, which is Flaviviridae, and because there is a similarities in them in some parts of the genome of both hepatitis C and dengue virus. So this will cause cross-reactivity and gives you a false positive. Immunocompromised patient or immunosuppressive, sometimes they may have a small or little antibody, so they will give you sometimes false negative. This is another problem with ELISA. For example, in hepatitis C virus, so if this person gets infected, HCV RNA is there, anti-HCV is there, but the problem is with hepatitis C virus, if this person get treated or get rid of the infection, and RNA will not be there anymore, but the problem with antibody, it will be there for ages. So when you do ELISA or even you use the confirmatory test, which is RIBA, still will get positive results. So this will misdiagnose or it will give false positive for this guy. So that's what happened exactly. So no more RNA is already vanished because of treatment, or this guy get rid of the infection by the immune system, but still anti-HCV is there. So this is another problem with ELISA. So after all these things, we were hoping to have a new technique that, make the life easy for us in diagnosing, or very sensitive, specific, and time, please. So thanks to this guy, Gary Morris, who invented PCR in 1993, and he got Nobel Prize for this invention. And PCR, for the people, I'm sure most of the people here, they are very familiar with PCR, but just as I mentioned, to have refreshment for the people coming after me and remind them about PCR. It's an amplification of fragment of DNA. So as simple as that. And we have two types of PCR, which is one is the qualitative PCR, which give you either positive or negative results, and the quantitative PCR, which is real-time PCR, which give you the amount or the exact copy number of this sample. Here simply is the stages of PCR, starting by the denaturation, where the double-stranded, they are separated from each other, and in annealing, where the primers, forward primer and reverse primers, attach to the complementary DNA, and they start to build the new double strand. And then this is the extension, where the new double strand are formed. So primer design, I'm sure this is one of the problems of PCR. So you have to be very, very careful when you design primers. Otherwise, you will end up with primer dimers in your reactions. So this is one of the problems of PCR. Although this kits comes from the companies and well designed, but still in some house assays, the places where they use in-house assay, they suffer from these things. We have two types of real-time PCR. We have cyber-green and we have Tagman. This cyber-green usually DNA binding dye, and this dye actually it binds to the groove of the double strand of DNA. So, but this binding of the dye to the groove of DNA, sometimes it will not be specific. It just binds to any double strand and emits when they bind. So this is the cyber green light. When the double strands are separated from each other, they will not bind because they usually bind to the double-stranded, to the groove of the double-stranded DNA. And then, when the double strands are formed, this dye will attach to the groove of the double-strand DNA, which give you the emission or the fluorescence or a sign of amplification. And sometimes it will not be specific because this probe just bind to any double strand. This is the same thing. So they are away or separate from each other, but when the double strand forms, this dye will attach to the double strand and they emit. And this is to make sure that your reaction is okay, otherwise you have to avoid the primer dimers for occurring here. As I mentioned before, the problem with the cyber green that the dye can attach to double strand or any double strand, non-specific attachment. And the problem sometimes in brighter primer dimers because sometimes they may amplify themselves. And another problem, which is major problem, you can't do multiplex in cyber green. So you have to check only for one virus. You can't do multiple primers to check for different viruses. Tagman overcome this problem, especially with non-specific binding. Now here the probe has nucleotides which can attach to the complementary DNA, which make the things more specific than the cyber green. So because here we have something, it's named reporter and quencher, which they attach to each other. So when this reporter, exposed to lights, it will emit. But this emission will be suppressed by the presence of the quencher. So this probe will attach to the single-strand DNA, to the complementary DNA, and when the primer started to form, or the new double strand, this attachment will be broken by the polymerase, which breaks here, and then it allows the reporter to emit freely, and this gives you signs of amplification. But we have a problems here. Although that this tagman overcome the non-specific binding, but still contamination is very, very annoying things. Although it's just a small word, but it's a big headache to the people working on PCR. And sometimes what happens for this, sometimes spontaneous break, although it's not frequent, but sometimes it can occur spontaneous break of the quencher and reporter, which may gives you a wrong or a false amplification. So this is another problem with the tagman. And it's labor intensive and expensive process and requires highly knowledgeable experience. This is our lab. Usually we have the dirty area where we extract the things, and then we move to the other area where we add primers and nucleosides, and then we move to this movement around. Actually, it caused a lot of headache to us, especially for contaminations. And in the last few weeks, we suffer a lot with this contamination. We cleaned everywhere, but still we are suffering because we don't know the source, or maybe one of the technicians, just falls down one of the samples and he don't want to talk. So this is the major problem we face in PCR is contamination, as I said, because you have to move around from place to place and to the third area where you settle, do the things. Thank you very much.
Thanks very much, Dr. Zaki. It's nice talk, by the way.
السؤال غير اللامة بلا يا جماعة.
Any question for Dr. Zaki? Okay. Now we have. Thanks very much, Dr. Zaki.
Okay, welcome.
Now we have